Okay, so topic 4, we're going to be talking about changes in the signal transduction pathway. As you know, there are 3 steps to our signal transduction pathway. First one is reception of the signal. Second step is transduction of the signal, which means it's amplified, sent to appropriate parts of the cell, and then of course you have a response by the cell. So let's go ahead and review and look at this model here and see what we've got going on. We have a growth inhibiting factor here, counter to a growth factor, which would tell the cell to divide, tell the cell to grow. This is a growth inhibiting factor telling a cell not to grow or not to divide. So you have reception occurring up here when this ligand binds to the receptor molecule right here. That is reception. Then you have these relay proteins in this area right here. This is all part of transduction. Um, so what we end up with at the end here is something called a transcription factor. Which is going to bind to our DNA and essentially tell our DNA, hey, I want to make this protein right here. And the DNA will respond in kind by making said protein. This protein that is made from this P53 gene right here, which is an important gene. We'll talk about this a little bit later more in depth. But this protein is a protein that inhibits cell division. So this would be considered the response, kind of the making of this protein. This is the cellular response. So if this is operating as normal and you get a growth inhibiting factor, this signaling pathway will occur. This transcription factor will bind to the DNA, and it will start pumping out this protein that inhibits cell division. So we want to know what happens when there is a change in this signal transduction pathway. Maybe this protein right here is mutated in this cell and it can't be made. It comes out with a different shape, right? Different secondary structure or tertiary structure. Uh, Same could be said over here. If we have maybe a protein that looks something like this, right? Then this transcription factor cannot bind to the DNA, right? Which means that everything else in this pathway, if you have any sort of a mutation here, this protein is made like this, then the rest of this won't happen. You won't get a protein that inhibits cell division, and what you might have is unchecked cell division, or a cell that just kind of constantly divides, and of course that's not good, right? So if that protein is absent, cell division would not be inhibited. You see, this is very common in cancer cells. You're going to see mutations in the P53 gene. This is also called the tumor suppressor gene, as I think you can guess why. Because it helps to suppress the uninhibited growth, unchecked growth of cells and the unchecked division of cells. So this is one example of how a change in a signal transduction pathway can affect things. There's obviously many examples of this. This is just kind of one I chose so that you can kind of visualize what might happen in this scenario. Okay, that's it for topic 4.4.